the introduction of life on Earth. And really, what we're going to focus on is taxonomy and how we classify organisms in nature that we find. So, when we talk about life, we first break it up into domains. And in my mind, I really break it up into prokaryotes and eukaryotes. But as you can see here, there's actually three different domains of living things. There's bacteria, archaea, and eukarya. These are the three main domains. And you can see here on the right, we've got a picture that breaks down each one. And what you'll notice about the domain bacteria and archaea is that visually they are similar. They're single-celled microscopic organisms. But there's some dramatic differences in their morphology and the way that they execute life that separates them. And so we have three domains. Now, of course, we belong to the domain eukarya. You are a eukaryote. If you see over here, we've got the kingdom Animalia. That's the kingdom you belong to. But eukaryotes are vastly diverse in their morphology. And it's very obvious that they're different. And in the other two domains, it's a little bit more nuanced, their difference. You have bacteria that live on and in you. So we talk about the domain bacteria. They're prevalent all throughout the world. And archaea are found in extreme environments. These kind of three phrases will help you break these up and distinguish between them in a very oversimplified way. If we talk about taxonomy itself, the phrase that comes to my mind that I was taught as a kid was King Philip came over for great soup. And so that's kingdom, phylum, class, order, family, genus, species. And it's an acronym to kind of help you remember all of those in order. As you can see here on the image, this is bears, and they're in the domain Eukarya. Their kingdom is Animalia. Their phylum is Chordata. Their class is Mammalia, and that's where our similarities differ. If you look at humans, we have all of those the same as well. But then our order is different. They're in the order Carnivora. We are not. They're in the family Ursidae. All those bears together. They're in the genus Ursus. And then, of course, here you have the American black bear, which is Ursus americanus. And that's a binomial naming system. So it's two different names. And if you take a look, it is the genus and the species. Sometimes you'll see trinomials where it's genus, species, and subspecies. So like dogs will have this. They'll be Canis lupus domesticus. Well, Canis is their genus, lupus is their species, and domesticus is their subspecies because they're a domesticated dog as compared to a wild dog. But as you can see in this picture, there's a lot of levels on how we classify an organism. So again, domain, kingdom, phylum, class, order, family, genus, species. So you could kind of turn my phrase that I was taught into a question. Did King Philip come over for great soup? Well, it'll help you remember all of these in order. So how do we actually classify this? What do scientists actually do? Well, first off, they look at morphology. They're going to observe organisms' physical characteristics, such as body shape, size, color, structure, etc. This is how Carlos Linnaeus actually started the naming system. And you can go back and find a quote for him that he did it for the greater glory of God. He wanted to name all the different species that God created and talk about their morphology. And that really is how it started. Things that looked similar were grouped together in this system. We also include behavior. So one of the things with an organism is their niche and how they live their life. And so behavior is really, really important. So like all the bears in Ursus are capable of hibernation. It's a behavior that they have, and that can help do it. Then we have physiology and biochemistry, so studies of the metabolic processes, enzymes and proteins and different stuff like that. So, if you look at different organisms, you can classify them by maybe what they can use as an energy source. Molecular data is in modern taxonomy and is a super powerful tool. And what it does is it looks at genetic analysis to compare the makeup of these organisms. And by looking at that genetic makeup, we've actually altered our classification of many species. Realize that taxonomy is a dynamic field. New data comes in, especially the molecular kind, and it actually changes how we classify organisms. For example, molecular analysis has led to significant changes in the classification of microorganisms by distinguishing archaea from bacteria and how their genes are set up. The next few slides are going to have a huge amount of information on them, and I'm not necessarily going to cover all of it. And so what you might end up doing for the guided notes is pausing and kind of reading through, because they're really dense descriptions about each of our domains. So our first one here is the domain bacteria, right? And they are prokaryotic. What that means, they do not have a membrane-bound nucleus and they don't have organelles. One of the things you'll notice about them that's unique is that their cell walls typically contain a structure called peptidoglycan. You can see it bold here. And peptidoglycan is comparable to cellulose in the cell walls of plants in what its function does, even though it's not comparable in its structure. And we can see other cell walls have a main subunit. But for bacteria, peptidoglycan is how their cell walls are made up. Bacteria reproduce through binary fission. There's a huge diversity in bacteria, and we often like to think that they're just all similar, but in actuality, there's probably as much diversity in the domain bacteria as there is in the domain eukarya. And so if we talk about their role in the ecosystem, they're so important in decomposition, nutrient cycling, and nitrogen fixation. Bacteria do so many things for the ecosystem. And actually, many of them are hugely beneficial for humans as well. And so having good gut bacteria or good bacteria flora on your skin can be really important. So domain bacteria, really diverse and lots of different stuff. If we take a look at the picture over here, you're going to see that they do not have a nucleus. You can see their bacterial chromosome grouped together in what's called a nucleoid. On the left, you see an artist's rendition. On the right, you can see an actual transmission electron microscope image. And it highlights some of the major structures that they have in their cells. Again, remember, these are very dense slides. You might have to pause to get all the information. Domain archaea, well, they're prokaryotes as well. They do not have a membrane-bound nucleus or organelles. But they actually are really kind of different. Number one, their cells don't contain peptidoglycan, which is your first indicator that they're not bacteria, because all bacteria have some type of cell wall that has peptidoglycan in it. They also have unique lipids, so ether-linked lipids instead of ester-linked lipids. This is really useful for them because where we find archaea are in some of the most extreme environments. And these changes in their lipids actually help them survive in these extreme environments. So we call them extremophiles. Some archaea are methanogens, producing methane as a byproduct of their metabolism. So archaea is a really unique grouping of cells that in general we don't spend a lot of time studying because their ecosystem interaction with us isn't as dramatic, but they definitely have a huge amount of diversity. Then we come to domain eukarya. And as you see the picture over here, you can see that I've got a plant, a couple animals, a fungus, an insect, all kinds. And so eukarya is what we think about when we think diversity. What actually eukarya means, or to be eukaryotic, is that you have membrane-bound organelles and you have a nucleus. So if we look at a eukaryotic cell, you're going to see they have a large nucleus. Later on in the course, we'll have a video that focuses on eukaryotic cells, and we're going to be taking a look at the different organelles that are inside there. They're typically larger, more complex than prokaryotic cells, and while eukaryotic cells can be unicellular, they're often multicellular, as you see in the picture to the right. There are multiple distinct kingdoms. So for me, I look at 4 different kingdoms: Protista, fungi, plantae, and animalia. These are the 4 major kingdoms that fall under the domain eukarya. And they play a wide role in the ecosystem. So many primary producers are there. These are plants that are actually producing energy-rich carbon molecules, typically through photosynthesis, and they're actually able to create all these sugars that actually support our ecosystem. There are consumers as well. This is predominantly ants. When you find consumers in plants, you're talking about like a Venus flytrap or something, they do that because they have nutrient-poor soil. So most consumers are actually animals or fungi. Fungi are indeed consumers. They break down organic matter. A lot of people think mushrooms are producers. They're not. But you can see that eukaryotes can be producers or consumers. And there's a huge amount of complexity in the domain eukarya. So if we summarize all this up in just short statements, bacteria are simple prokaryotic cells. They're found everywhere, and they're absolutely vital in ecological processes. Archaea are prokaryotic organisms, often extremophiles with unique biochemistry. They're not as proliferant as bacteria, so we don't see them everywhere. And then eukarya are complex eukaryotic organisms. Remember that means they have a nucleus, including plants, animals, fungi, and protists. And they're capable of forming multicellular structures. So one last thing to address is the idea of why cells are small. Because we talked about all of these domains, and in archaea and bacteria, they're single cellular. And eukaryotes have single cells as well, but they tend to group up into multicellular organisms that are made of tiny cells. The short answer for why cells are so small instead of a giant cell is that diffusion is free work for small cells and it's not for large cells. Your cells rely on diffusion to help move molecules from area of high concentration to low concentration. In the future in the course, we'll have a video on diffusion that'll go in depth. But the bottom line is, if we were one giant cell, diffusion struggles to accomplish all the tasks that we need. Realize this is a general rule. Some exceptions do exist. So one thing I'd like you to do is do a quick Google search on this particular Calpera taxifolia and see one of the exceptions where we have an organism that has exceedingly large cells. But when we look at us and why eukaryotes are multicellular, in general, it's because diffusion is free for small cells and not for large cells.